We're going to start the meeting in a couple seconds there.
Hospital in
Unmute
Washington,
everyone.
a big cancer one.
Hi, Gina.
Hi.
Okay, hold on. Yeah, Amanda's okay, good. All right, so welcome everyone to a ALK Talk with Dr. Lam. And I'm going to open up by saying Wednesday, Cinco de Mayo, join us. We're going to have a great time, and that's 8 o'clock Eastern time. And I'm going to open it up with some poll questions, as I always do. So the first one, I'm going to launch the poll. What type of treatment are you currently taking? And I'm going to be talking out loud because we're recording. So this does not get recorded. So I have to actually give the results.
I think that's always so exciting to see people still on alectinib. That's really exciting stuff. That gives us all a lot of hope.
Hey, can you do me a favor? Can you just watch the waiting room? We
Yeah,
got some
I got it.
people that are entering
On it.
later. All right, so I'm going to end that, and you can see alectinib is 68%. Second, actually, is brenactinib, which is interesting, 13%. Lorlatinib's 11%. So I'm going to move to the next polling question.
Oh, did you share the results of that, Mark?
Oh,
You want me to do
geez,
that?
sorry. Okay,
There
let me
you
go
go.
back.
We'll let everybody
All right.
see it.
All right. So I'm going to stop sharing. And I'm going to go to my second question and launch this one. What's your length of diagnosis?
We got a nice mix. Some newbies, some people
Yes.
here of 1 year and 4 year. That's exciting.
Yeah. Gina, you want to announce it to the recording?
Oh, sure. Here we go. So it looks like I'm gonna, oh, did I share that?
Oh, I did it.
There we
Go
go.
ahead.
Oh, we were both doing it. So it looks like 24% are newbies, 1 year or under, and 30% are 1 to 2 years. 29% are 3 to 4 years, 8% 5 to 6 years, that goes above, and 10% 7 to 8 years. So that's pretty exciting stuff. If you're not on mute, can you mute yourself? And if you have any questions, you can ask them in the chat.
Okay, I'm going to launch this one. How many TKIs have you taken? And also, this is multiple choice. You can also add chemotherapy or immunotherapy. Okay, I'm going to end this poll. Share the results. 63% of us have only taken 1 TKI. 2 TKIs follow with, actually, as traditional chemo also has been added for 23% of us. 21% has 2 TKIs. And then we also jumped down to 3 TKIs, which is 8%. So I'm going to go ahead and go ahead to the fourth question. Should be an easy one. What stage were you at diagnosis?
And it sounds like somebody with a bird might be unmuted. So if you have a bird, mute yourself.
That or come back inside. We're going to end the polling, and there we go. 86% of us were staged at stage 4 and 11% stage 3. So I'm going to go to the final question. Okay, so this one is, if you have progressed, what are the areas of progression? You can do multiple choices, and if you have no progression, there's one at the bottom. All right. We're gonna probably be all over the map, but I can tell you. You guys see that? Gina, can you see that?
Yeah, well, yeah, but I can anyway. So I think everybody
yeah.
else can.
Okay, so no progression is 52% of us have not had any progression, which I guess makes sense because most of you guys are on alectinib. But you have lung, brain, lymph nodes, those are about the same at 17, 15%. So we're kind of all over the map on that one. So those are my questions. Cinco de Mayo is Wednesday, and I'm going to go ahead, 8 o'clock Eastern time, be there. I'm going to flip the microphone over to Gina with a couple of other announcements. Go ahead, Gina.
Hi, everybody. It's so good to see you guys on Sunday. My name is Gina Hollenbeck, and I'm the president of ALK-positive Incorporated, which was an organization that was formed out of the membership of the ALK-positive support group. We are all volunteers. We are patients and caregivers working together to improve the life expectancy and quality of life of ALK-positive patients worldwide. And so we're so excited to be able to offer so many things. But right now we've got a few events going on and we need your help because we know that as we work together as a patient community, that's how we are stronger and that's how we're better. And that's how we've been able to fund 2 clinical trials, some amazing research, and make sure that ALK-positive research is on the map and getting funded. So we have our Walk Positive Challenge going on through the entire month of May. We hope that you guys can join us in that. If you the challenge is basically walking 10,000 steps in 10 days, and you can get some people to sponsor you to help fund some amazing research. The other amazing thing that we have going on right now is we actually became our own nonprofit, which we're super excited about. So as much money as possible goes to research for us to be able to run this organization and have this research funded for ALK-positive cancer. Another amazing thing, there's Stephanie Peace put up the hashtag, which is Walk Positive Challenge. Another amazing thing is our ALK Summit coming up. And so I'm going to hand it over to Amanda to let her talk to you about how incredible it was. She was our our ALK Summit chair in 2019. And she has some incredible things to say. She did an amazing job. So go ahead, Amanda.
Hello, I am Amanda Nerstad. I am also an ALK patient. I was diagnosed in 2016. And so I'm going to do our shameless plug. I am ALK Summit Chair for 2021. And please save the date, July 31st through and August 1st is our virtual ALK Summit. Registration is coming soon. And we're going to have some great doctors, researchers, speakers. Dr. Lam is also going to be there, so we're excited about that. So that's my shameless plug. Save the date. But we are excited. We're honored to have Dr. Vincent Lam tonight. He's got some great research in the works, and I'm excited for him to tell everyone about it and about his vaccine. So I will let him introduce himself. Dr. Vincent Lam is a researcher and oncologist at Johns Hopkins. And thank you for being here, Dr. Lam. And if you could just introduce yourself, but also I think it's really cool about Dr. Lam, a little bit about his background and how and why you became a researcher and oncologist. So I'll let you take it away.
Oh, I didn't realize we were going to go into my background. I am so excited to join your conversation tonight on a Sunday evening. I'm scrolling through 5 pages of people on the Zoom call, and it's just amazing to see, you know, how committed and just engaged you all are. And that's, you know, a big reason why I focus on ALK lung cancer. So it's great. Thank you for having me. And you know, I'm seeing some familiar faces too, so it's wonderful. I just a little bit about myself, I guess. I am from the West Coast, grew up in Seattle, and started my career as a computer science major and a software engineer. I worked in Silicon Valley for almost 5 years. And then for some reason had this epiphany that I needed to do medicine. And so I did my medical school at San Francisco, and then ended up coming to the East Coast for training in oncology. My first faculty job was actually at MD Anderson in Houston, where I met Amanda and some of you all on this call. But I'm really, really pleased to be able to get back to the East Coast. And so I'm at Hopkins, where I focus on lung cancer. I also lead the esophageal cancer research program here at Hopkins, because at Hopkins in the thoracic oncology group, we see both lung cancer and esophageal cancer.
Great. So the one thing I love about Dr. Lam is that he's very positive. So for his patients that are on here, he's always very positive, and he's very proactive at the approach that he goes with his, that he does with his patients. So you're doing lots of ALK research, and we're so excited that you are. So could you tell us a little bit about specifically, can we start off by asking, lately you've been testing your ALK patients' blood. So what is the research that you're doing with ALK blood recently?
Well, we're trying to do several things, actually. So, first and foremost, our main focus, my main focus, really at Hopkins is to leverage our expertise at Hopkins, which is immunotherapy. So our areas of research are really focused towards trying to figure out, a few main things. One, why ALK doesn't respond to immunotherapy as well. So really trying to lay out what they call the tumor microenvironment and other factors involved in your immune response to ALK, and to really try to sort out the specific reasons why immunotherapy just doesn't seem to work as well in this particular disease. And then on the therapeutic side, our efforts are trying to develop novel immunotherapies, because as you all know well, the current immunotherapies such as Keytruda and Opdivo really don't respond very well, or don't treat ALK very well at all. And so we feel that there's really just a strong need to figure out better treatments that can better engage the immune system to get us to where we really want to go in terms of treating ALK, and that's turning it into this chronic, long-term disease, if not a cure.
Perfect. So there's a group of us that are fundraising for you for your vaccine, and we are really close to getting it funded for your phase 1 clinical trial. And we're really excited because we feel that it does and will have an impact on many of us. Can you talk and describe about your specific ALK vaccine and to tell us about that?
Yeah, so one of the novel immunotherapy strategies that we're looking at and that we've been working on for some time is really vaccines. So taking a small step back, at MD Anderson, I led T-cell therapy trials for lung cancer. And so I really have a strong focus and interest in trying to figure out different ways to introduce these newer immunotherapies to an immune cold tumor like ALK is. So vaccines is one strategy, and so we're very excited about this potential treatment. But we're still in the early stages, which is why I'm so grateful for Amanda and a lot of our other patients who have really taken it upon themselves to help us get the funding to really get going with this project. But just very briefly, vaccines are really meant to try to really teach your immune system about the cancer itself, just like it's been used to teach the immune system about COVID and other infectious diseases. So leveraging that paradigm, we have been working on a vaccine in ALK, but a slightly different twist than what Dr. Awad and Dr. Chiarle are doing an amazing job with their project. We are really trying to develop a possible vaccine for the huge unmet need of ALK patients who are actually doing well on their TKI, but unfortunately do feel that they're sort of sitting on a ticking time bomb, as some of you have described to me, because we do know that unfortunately acquired resistance and progression on a TKI does happen more often than not. So with our vaccine approach, we are really trying to potentially leverage this novel immunotherapy for patients in that situation.
That's great. So can you describe a little more in detail how your vaccine is complementary and how it is different from other ALK vaccines that are in the works?
Yeah, so if I take a sort of a step back in terms of just introducing the concept of vaccines, and I know Dr. Awad and Dr. Chiarle have talked to the group, so you have some knowledge about this already. But cancer vaccines are not new. It's been, the field has been developed over the past, few decades. And of late, actually, particularly in melanoma, there have been some really exciting advances in terms of using a vaccine to, as I mentioned earlier, to educate your immune system about your cancer, so that it can just serve as a different way to basically wake up your immune system and say, hey, you need to take care of this cancer. And so, unfortunately, we haven't made a ton of progress with cancer vaccines, although, as I just mentioned, of late, there's been some really exciting developments, particularly in melanoma. And the reason why we haven't made a ton of progress is because as you can imagine, it's an extremely hard problem, and it's a hard proposition to present the proper information to your immune system through a vaccine, and then set it up such that all of the cascade of immune events that are needed to affect a substantial immune response happen, such that the tumor starts shrinking and things like that. And so our solution really is aimed at trying to take a smaller bite of that apple, I guess, and really to try to circumvent some of the issues and challenges that are involved with building a vaccine for the progressive metastatic setting, which is, a vaccine for patients who are stage 4 and, are progressing on a TKI or chemotherapy. And so that is the setting in which most vaccines have been studied in the past. And as I just mentioned, that's a difficult proposition, although really exciting progress by, like groups like Dr. Awad and Chiarle. We are trying to circumvent some of those challenges by bringing the vaccine earlier in the patient's cancer journey. Because we know, through all of the cellular therapy and vaccine work that's been done, we've learned a lot of important lessons, and that's why I'm really optimistic that we're going to get there in terms of either a T-cell therapy or a vaccine for lung cancer and other cancers and for ALK. But one of the lessons that has been very consistent is that immunotherapy works best when there's not a lot of tumor. It can work when there's a lot of tumor, but it works best when there's minimal tumor burden. The other thing we've learned is that you know, the immune system can actually ignore signals from that are presented to it, let's say from the tumor, and often because it thinks that it looks too much like normal healthy tissue, because obviously you don't want an autoimmune process. And so that's one of the hypotheses about why potentially ALK may not be as immunogenic as some of the other tumor cancer types like melanoma. Melanoma is very immunogenic. Your immune system in general goes crazy when it sees melanoma. And the hypothesis is that ALK actually looks too close to normal tissue potentially. And so, you know, with moving the vaccine up closer, up earlier in the patient's cancer journey, we're hopeful that, and we're optimistic that we can actually solve a couple, or at least avoid a couple of these main issues that have been holding back, you know, vaccines in the progressive metastatic setting. So, number one, if we treat people who are doing well on their current TKI and have very little disease, if at all, if any disease, because many are NED, fortunately, then that takes care of the issue of having to, you know, rely or generate such a robust immune response that it has to, you know, shrink a ton of tumor. Number two, because interestingly, your immune cells actually get exhausted. There's an exhausted phenotype that's well described. And so that probably plays a role in why immunotherapy doesn't work as well when there's a lot of tumor. Number two, the novel thing that we're doing with our vaccine that to the best of my knowledge has not been done before is we are actually using the vaccine in a prophylactic sense and not a therapeutic sense. So the distinction is that what we are putting in the vaccine and what we're trying to teach the immune system about is actually something that you shouldn't have yet. So again, drawing upon the successful experience of, you know, our infectious disease vaccines, for coronavirus and the like. So our proposal and what we're working on is really to actually vaccinate patients who are doing well under TKI against and have not had any acquired resistance. So we're going to actually teach the immune system about the acquired resistance mutations beforehand, before patients attain these resistance mutations. And with our vaccine, you know, the specific mutations that were included, it looks like we'll be able to capture maybe around 80 to 90, probably 90% of the acquired resistance mutations that are seen after alectinib or after brigatinib, like G1202R is a common one. And so that's, in a nutshell, that's what we're trying to do, is to actually educate your immune system about these bad mutations in advance, so that hopefully if the TKI starts, you know, breeding these resistance mutations, your immune system has memory about it because it's seen that pattern before from the vaccine and springs into action to keep it down. And hopefully that means that you can stay on your alectinib or brigatinib for a longer period of time.
So you're hoping that we can get the vaccine earlier. And what are the requirements for your vaccine? Is it to be NED? Is it to be stable? What are your requirements for that?
The requirements, so again, I want to stress that we're still really in the early stages. We have some, you know, what is called preclinical data. So data in test tubes, I guess, for lack of a better term, to really show that this approach really is immunogenic. It really does elicit a really strong immune response. So we are not, so the one thing I guess I should comment on is the clinical trial aspect of this, because we are still relatively early in our development of this approach. As you probably know, commonly how these clinical trials come about with novel therapeutic products is oftentimes you have these preclinical data, which often include mouse studies and such. And then once you see those results, then you open a clinical trial. And we are, in the process, as I mentioned, of continuing to develop that preclinical data. But one of the nice things about our vaccine product is that it's actually been used in sort of the construct, the overall general construct of the vaccine has been used in multiple studies previously and has demonstrated to be quite safe. And then the exact vaccine product that we're proposing in terms of the main components are actually in a clinical trial right now with my colleague, Dr. Mark Yarchwan, for liver cancer at Hopkins. So we have a lot of confidence that the vaccine itself is safe, that safety is not an issue. So therefore, along with my senior co-collaborator, Dr. Liz Jaffe, who's been working on vaccines for decades, we strongly believe that this presents a really nice opportunity to actually bring it to the clinic at the same time as we are working on the mice studies. Because as you may know, we have made some really, the field has made some really amazing improvements in terms of the mice models. But there still is not a good model that can truly recapitulate what the human immune system is gonna do with this vaccine. So we strongly feel that the only way to see whether or not this is going to work, and if not, where does it get hung up in that whole cascade of required events to really make it work, we have to put it in people, and that's how we can actually accelerate the development of vaccines and other cellular therapies. And so just to quickly go back to, I guess, your initial question about how it may be complementary to Dr. Awad and Chiarle's vaccine, I think that the data that we generate from putting it into patients quickly and the comprehensive scientific analyses that we'll do on the biospecimens of patients who are in the trial will help inform all cellular therapy and vaccine efforts, and hopefully, we'll be able to help facilitate Dr. Awad and Chiarle's efforts as well. And any information or data that they get from their efforts definitely would be helpful for us as well. So it's complementary in that way, and then it's also complementary in that they're aimed at patients at different parts of their cancer journey.
That's
You
great.
have
So
some great questions.
Yeah.
I know. So we have some great questions. We will start to go in the chat. Does this vaccine from Stephanie, does it cover the blood-brain barrier?
That's a great question. We don't know. And so that's why I hopefully you won't get sick of me saying that, but that's the honest truth, for a lot of these questions. And that's why we do feel very strongly that in order for us to really know, we have to start putting it into people if we have a product that we believe is safe. So vaccines in general should, based on previous mice data and stuff, it should be able to address, disease in the CNS, but we don't know for sure in humans.
So we're gonna skip the mouse phase and get the results from people.
Yeah. I mean, again, not so much skip the mice phase, but to do it in parallel, because as you know, Gina, to do mice studies, it'll actually take a couple years.
Yeah,
you know.
yeah,
So
and
we
that's
can do
time
it
that
together.
lots of us don't. Yeah, that's time lots of us don't have.
Yep, and then my studies are so critical because it'll help us optimize the vaccine in terms of dosing and other kinetics and things like that.
So
Piggyback.
can you,
Oh,
oh,
go ahead,
sorry.
Amanda.
Can you describe a little bit more? You said that, immunotherapy is known not to help ALK, but this vaccine is an immunotherapy. So how does that, can you describe and talk a little bit more about that?
Yeah, so I was sort of just colloquially referring to immunotherapy in the sense that, sort of referring to the immunotherapies that are currently FDA approved in the US. So that is specifically immunotherapies like Keytruda and Opdivo. So those are immune checkpoint inhibitors. Immunotherapy that really aims to release the brakes on the immune system that tumors put on your immune system, and those brakes are called PD-L1, which some of you may be familiar with. And I bet some of you have seen some of your pathology reports, and it says PD-L1 like 100% or 90%, and maybe some of your oncologists, have been tempted to actually start Keytruda or Opdivo, for that, but. And so that's what I'm generally referring to, these immune checkpoint inhibitors that are currently available. We know through retrospective studies and registries that the response rate for these types of immune checkpoint inhibitors in ALK-positive non-small cell lung cancer is very close to zero, if not zero itself. So we don't use immune checkpoint inhibitors in ALK non-small cell lung cancer. Of course, there have been studies that have been done to combine them with TKIs, and toxicity have been an issue. So that's why it's really not a standard of care at this point. Oh, and the second part of your question is, how is the vaccine different? Well, the vaccine is different because immunotherapy is such a broad umbrella. It basically refers to any treatment that can enhance your immune response to ALK or any cancer. And so the vaccine is different because it just tries to enhance your immune system's awareness about ALK, about your ALK cancer in a much, much different way.
Gotcha. Here's a question from Wendy. Many of us had significant tumor burden at time of diagnosis. Would your recommendation then be to shrink tumors with TKI first and then administer the vaccine?
Absolutely. And again, we don't have any ALK vaccine experience in people, to really definitively guide us. That's what we're trying to do. But based on, other cellular therapy and vaccine studies, I strongly believe that these novel immunotherapy strategies are best deployed in the context of minimal tumor burden. And in fact, of note, the melanoma studies, at least one of the melanoma studies that have actually shown really promising results they've only treated, I think, 8 patients or so. But 4 years out, many of those patients are still doing relatively well, i.e. no recurrence. And they actually just published a paper not too long ago that actually showed that they were able to demonstrate memory T cells in those patients that came about from the vaccine that they were given. But the notable thing about that study is that it was done in patients in the adjuvant setting, meaning these melanoma patients actually got surgery. They were not generally not distant metastatic stage 4. And so they got surgery for their melanoma, and then, as a curative approach, and then got the vaccine as sort of an insurance policy, so to speak.
Gotcha. And here's another question from Stephanie. How would you know then if you qualify for the vaccine?
So I'm interpreting that to mean, what the eligibility criteria might be for our clinical trial. And again, we're still working out the details of the clinical trial because as Amanda noted earlier, we were still trying to get this funded. So we actually, it's still not funded, quite yet, but with the heroic efforts of Amanda and others, we're really getting close. So I don't have the protocol fully worked out. We're working on it right now, but I'll tell you what I'm thinking, what I'm initially thinking the eligibility will be though. It'll be for patients who are on their TKI. It doesn't have to be first line, it can be any line. Who basically actually, and doesn't even have to be TKI, but for patients who are on TKI or chemotherapy and have had stable disease that has been demonstrated to be stable for at least 4 months or so. We haven't quite set that in stone yet. And that's about it. You don't have to have measurable disease. You can be NED, in fact. Would be even better if you were NED, because we are not treating active progression, so we don't really have to measure what your disease progression actually is in terms of scans.
Perfect. And here's a message from Julie. What about resistance that develops not from acquired mutations but from bypass mechanism? Will the vaccine work for that situation? About 50% of all progression.
That's a great question. And so no, that the bypass mechanisms, the ALK independent, progression remains a key issue, for our ALK patients. So the vaccine doesn't directly address that. And as Julie astutely notes, it really would only cover about half of the expected acquired resistance, for a patient on alectinib or brigatinib, for example. But an interesting thing about vaccines, which is why, I so strongly feel that we really have to get more of this research in terms of how to better engage the immune system, because it's more of a live drug, not, it doesn't have this sort of the static activity as, for instance, a small molecule like a TKI has, which has been invaluable in our efforts against ALK. But to combat that, the trickiness of this darn cancer and how it, gets acquired resistance, the vaccines and cellular therapies like vaccines actually have this concept and this potential of what's called epitope spreading. So meaning that even though you're not directly targeting a part of the tumor, over time, through the process of the immune system responding to the part of the tumor that you are targeting, it can actually learn about other parts of the tumor that you did not initially teach it about. So in fact, again, this melanoma study that I've been mentioning, that was one of the key things that they were able to demonstrate at that 4-year or so mark, and that indeed some of those patients had evidence of epitope spreading, in that their T cells now recognize parts of their tumor that they did not get originally vaccinated for.
How about people on lorlatinib that are stable on it? Do they meet your criteria for the vaccine?
Mm-hmm. Yeah, the only other, I guess I forgot to note in my response about the eligibility, the other major eligibility requirement that we would have is that you can't have one of the, number of acquired resistance mutations like G1202R that we're vaccinating against. So, if you're on lorlatinib, chances are pretty good, unfortunately, that you do have one of these acquired resistance mutations, but it's, there's a small minority of patients that may not. And so those patients would be eligible as well. And the reason why we're doing this is because while we do, again, hope that there may be some epitope spreading and the vaccine will be able to cover parts of the tumor that are not being vaccinated against, as a pilot study, we really have to be very crisp about the hypotheses and how we're measuring success. And so limiting it to this very narrow scenario of patients who don't have what we're vaccinating against, and then seeing upon disease progression if it actually worked. In other words, was the progression related to any of the things that we vaccinated against? That I think will help push the field a lot, forward a lot faster.
So it's hopefully giving the vaccine before you get the G1202R, before we get that, so you can hopefully never get it.
Right, but that's where it's amazing that, for instance, ALK-positive has funded this amazing effort by Dr. Awad and Chiarli. You can get on their vaccine trial. I want to pause for just a second and note how amazing it is for ALK to actually have two, bona fide vaccine efforts, underway. I don't actually think even EGFR has, sort of a setup like that right now. So, I think it's just a testament to, to you guys.
Also, yes, because we all fundraise for ALK specific, treatment plans. And that's so great that there are, an earlier disease vaccine that you're doing, and we're hopeful for Dr. Awad's vaccine too for later, for those later mutations, which is great. How do you know if the vaccine stops responding, or how do you know if it worked or not?
So, in contrast to the setting where we're vaccinating patients who are progressing with, in the metastatic progression setting, in this setting, our measure of success initially is it's sort of iterative. So the first milestone and what we're hoping to see and what we'll be measuring is checking in the patient's blood, patients who've been vaccinated, checking to make sure, to see if we see an immune response. So looking at the particular types of T cells that have arisen, checking specific levels of cytokines or chemicals that your immune system releases when it gets activated. So that's really sort of the first measure of success. Does the vaccine actually do in humans what we actually see it do in the lab, which is that it actually gets the T cells pretty excited. And then secondarily, the clinical measures of success are then looked at, and that includes, for the patients who are vaccinated, do they stay on their first-line TKI longer than what we would normally expect? And also, for instance, if when they progress on the clinical trial, what does the progression look like? The molecular profile of the progression look like? Did they progress with G1202R that we vaccinated against? Or was it all bypass mechanisms? So those are sort of the basic measures that we're initially looking at.
Great. And this is Jim is asking, why vaccinate specifically against ALK resistance mutations rather than all ALK protein in general?
That's a wonderful question. That'll be probably step 2. And again, science is an iterative process, and and there's a nice, there's a fine balance that I'm trying to achieve in terms of trying to ask a focused, narrow question that can move the field forward as quickly as possible and to generate data to share with other groups that may be working on similar things. And so, as I mentioned earlier, I think not the easiest, but the quickest way and the cleanest way to learn about how this is going to work in humans, how well it's going to work in humans, is to actually focus the question to acquired resistance.
That's great.
But that's a great, sorry, Amanda, but that's an intuitive next step, because along the lines of what's been done in other disease groups, we should vaccinate against ALK wild type is what it's called, without the acquired resistance. So, but one reason why this is a really focused and narrow question towards acquired resistance is, I had mentioned this earlier in terms of the challenges for vaccine strategies that have come before us. And so vaccinating against acquired resistance mutations is really appealing because, theoretically, this is a completely foreign thing to your immune system. You should not have G1202R, for example. So we have some data that show that actually is more immunogenic than vaccinating for ALK, the ALK fusion protein itself, because sort of a basic way to look at it might be your immune system has likely seen your ALK fusion protein before, because there's a reason why it became metastatic, right? So your immune system either hasn't seen it at all, which is, there's data that's been generated by other groups that says that's less likely the case. But so if it's seen it, it's decided not to do anything about it, or it's unable to mount a significant response to it. So it just complicates the question a little bit more. And I feel that if we can actually demonstrate this in a clinical trial for a highly immunogenic sequence, that would be a great starting point as a proof of concept.
That's great. And it seems like I think some questions were answered on this, but can you describe the difference? So for your vaccine, they need to be stable for 4 months or NED. What is the difference between stable and NED? Can you talk about that?
Oh, stable just means, so the other term you might hear is disease control. So it just means that you're not progressing. Yeah, so it could be that you have zero response to your first line TKI, but as long as it's not getting worse, then we're good. Yeah.
It's not growing.
Yep. And NED, colloquially refers to the fact that there's no overt tumor, on your scans.
Great.
I think there's an overall question just that keeps happening, that keeps coming up, and it's just that, would this go to other cancers, or would it even be applied to ROS1 since it's so close to ALK?
Yeah, and that's why we're really excited about it. And again, to my knowledge, I don't think anybody's done a prophylactic approach. And so you can imagine that so ROS1 has a very similar acquired resistance landscape, as an analog really to ALK. And so, if we can demonstrate that this works, I think it would actually apply to potentially many other disease types, and even outside of lung cancer.
Great. And from JT, do you see the vaccine strategy evolving to leveraging mRNA as a delivery method?
Yeah. So for the broader audience, there's different ways to deliver the information that you want to the immune system via a vaccine. So there's peptides, which is what we're using, and our group has a lot of experience with. There's DNA, and then there's mRNA. the COVID vaccine, Moderna and Pfizer fame. So we don't, there's technical differences between the three. We don't really have any clear, evidence one where, that really suggests that one is better than the other at this point. The nice thing about peptide is that, well, there's many advantages to using peptides, and I believe that's what Dr. Awad and Charlie are using for theirs as well. They had also looked at DNA vaccines, I believe. So there's many advantages to using peptides.
That's great. Dr. Lam, as you know, most of us on this call have ALK, and we know how serious our disease is. Is there anything that you can help provide hope for us for that you think is coming down the pipeline in the next few years that can help us?
Yeah, as I mentioned earlier, I'm really optimistic that we're going to make some significant headway in the novel immunotherapy space, and that includes vaccines, T-cell therapies, and the like. There's different ways to approach treating ALK, and I firmly feel that to get to where we really need to go in terms of achieving a long-term durable response in ALK, while we're grateful for the TKIs and small molecules and stuff, and we will continue to need to lean on those, to get to where we eventually want to go with this disease, we're going to have to find a way to better engage the immune system. So the good news is on that front, there's progress being made. And the nice thing is ALK is not responsive to the current immune checkpoint inhibitors. And so that's a very difficult nut to crack. But the nice thing is a lot of lung cancer doesn't respond to the current immune checkpoint inhibitors. So it's not a new issue that the field hasn't been grappling with for the better part of this past decade almost. And so we can leverage in ALK a lot of the progress that's already being made in non-small cell lung cancer that's immune cold, such as ALK is. And then in terms of non-immunotherapy strategies, these combination therapies like the ALK plus SHP2, TKI plus SHP2, in combinations, or MEK, those are just, they're great, and those will really help, are already helping us push the ball forward, until we can get to a really long-term sustainable immunotherapy strategy. And then, of course, sort of farther down the horizon, but not impossible, really, there's been some progress made in the whole protein degrader area of drugs. So instead of a TKI to stop the activity that's downstream of this fusion protein that's causing all these issues, a drug that actually just goes and tags your fusion protein for disposal, basically, from your, by your cell's normal garbage collecting mechanism. There are a lot of challenges still in that space, but there are already companies that are working on similar things for, RET, for example.
So for those that are progressing on lorlatinib, are there any clinical trials that look promising for immediate need that you can see? You kind of described it just now, but anything else that you can recommend that you tell your patients or help your patients with that are progressing off of lorlatinib?
Yeah, that's the the sort of $1 million question currently. What to do, after lorlatinib. Thankfully, we have post-lorlatinib drugs coming. So TPX 0131, a more potent TKI that can address some of the compound resistance mutations that happen as a result of lorlatinib. There's another more potent TKI along the same veins, along the same vein, Nuvalent is the company that, so there's more potent TKI, but that's not quite ready yet for clinical trial enrollment. What's ready right now, I would say really, I'm very enthusiastic about Dr. Degago-Jack and colleagues' combination trial combining, lorlatinib with MEK inhibitor or SHP2. We have MEK SHP2 combinations, although not specifically for ALK. And I've had patients tolerate that fairly well and have done well on that. So I think that would be my sort of go-to trial right now. And then outside of TKI specific trials, we've seen some interesting, some results in our, not ALK specific, but EGFR as well. So this is a non-ALK specific trial, but there's a class of drugs called TROP2 inhibitors or antibodies. And there's actually several of them that are in development, but but at least a couple of them are actively enrolling in clinical studies now. So these TROP2 inhibitors are actually, I've actually had some patients who are, EGFR or ALK, primarily EGFR, who have responded to TROP2 inhibitors on these trials. So I actually think that may be something that's worth looking into, for a non-ALK specific option. And then finally, a little bit more of a stretch because we're not quite there yet in terms of cellular therapies. But as you know, Amanda, at MD Anderson, we did a lot of cellular therapy studies. And so, that field is really making a lot of progress. They've seen some initial results already for gastroesophageal cancer. And so, that's something to be considered that's readily available in, major academic centers. And that could be an option.
You recently said that when TPX-0131 does open up for clinical trials, that Johns Hopkins will possibly be one of the hospitals you're the point person for Johns Hopkins.
Mm-hmm.
Can you elaborate on what you know about TPX-0131?
Yeah, so TPX-0131 is the next potent TKI that we're hoping that that can help our ALK patients, and specifically in the post-lorlatinib setting. So it specifically has been demonstrated to be effective in preclinical data against the compound mutations that are that cause resistance to lorlatinib. So these combination solvent front and gatekeeper mutations, for example. And so we're quite excited about that. In fact, I think it is starting, early phase clinical trials in Australia now or soon. And they'll be bringing it, starting these trials in the US, hopefully by the end of this year.
That's great. Well, we want to be respectful of your time. Is there anything else that you want to tell us or ALK patients?
No, thank you so much for taking time out of your Sunday to talk ALK science and clinical trials. I'm just really I feel so honored to be part of your community, and please don't hesitate to reach out if you have any questions at all.
Awesome. Well, we thank you so much, and this is great information. We're all excited to move forward with hope on the horizon for sure, and all the research going on for ALK. And if you look, and everybody could please give a take off mute, and Mark does this usually, and say thank you to Dr. Lam for his time and all the research.
Thank you.
Thank you. Thank you
Dr.
very
Lam,
much.
thank you for all you do for us.
Thank you very much.
Good doctor. Thank you.
Thank you, Dr. Lam.
Take care, guys.
Good luck.
Thanks, Dr. Lam.
Bye.
So good to see everybody.
Thanks, Dr. Lam.
Thank you. Thank you, Dr. Lam.
Bye, Amanda. Bye, Gina. Bye.